So good afternoon, everyone. Good afternoon, good morning, good evening, depending on where you are connecting from. And welcome to the third Global Neuroscience Horizon webinar organized by IBRO. I'm really glad to be here today with you. We have a really interesting and fascinating topic. The topic of today's webinar will be the links between neuroscience and COVID-19, which is a very interesting topic. Still a current one, unfortunately. We were discussing before starting this event with our speakers that when we selected this topic a few months ago, we all were hoping that today, we would be talking about something that has been already relegated to the recent past. Unfortunately, COVID-19 is still with us, it's still making the news, which on the other hand makes this today's seminar even more interesting about a topic which is very much debated. So we were thinking that it would be interesting and important also for young neuroscientists to be informed about what we know, about what we still do not know about the ability of SARS-CoV-2 to actually invade the brain, to trigger neuroinflammation, to trigger a cascade of neurological and cognitive consequences. And for this reason, we have three fantastic speakers today with us. We have Alexei Verkhratsky from the University of Manchester. We have, Alexei talk will be about the role of glia, neuroinflammation in neuropsychiatric and neurological aspects of COVID-19. We will have Roxana Carare. She's a professor of clinical neuroanatomy, equality and diversity lead at the University of Southampton. I forgot to say that Alexei Verkhratsky is a professor of neurophysiology at Manchester University. And then we have Livio Provenzi. He's a senior researcher of the Mondino Foundation, which is the place where I'm speaking to you from, in Pavia. It's a National Institute of Neurology. And so we will cover three topics. We will cover about the role of glia neuroinflammation triggered by COVID-19. We'll be discussing about the pathological consequences of SARS-CoV-2 brain invasion, but we will also be discussing about the indirect consequences of these pandemics on mental health, the consequences that are related to the social restrictions that this pandemic has imposed to our lives. So we will have a little change in the program because I was just informed that Roxana Carare, she's having some little problems with the connection. So Alexei Verkhratsky will be the first one to open up this fantastic meeting. So Alex, please, the stand is yours.
Thank you very much. And of course, naturally, my first words of gratitude goes to IBRO. Thank you very much for giving us platform, and goes to Fabio, because this is really, I think, incredibly timely thing to talk about neuroscientific or neuroscience, neuropathological consequences of this particular pandemic. Now I will try to share my screen. And I hope it works. Do you see it? Yes. So I'm going to talk about neuroglia in neuropsychiatric and neurological aspects of COVID-19. And as you remember, Fabio, when introducing my lecture, also added the word neuroinflammation. In fact, I deliberately removed this word of neuroinflammation because this is something which is extremely important to define. And for the time being, we don't have a definition of what neuroinflammation is. And that may have clear therapeutical consequences, because if we think about chronic neuroinflammation as a reality, then we try to, of course, cure it with anti-neuroinflammatory agents and nothing happens. And that already we have seen in Alzheimer's disease and neurodegenerative diseases, because chronic neuroinflammation theory of which actually spectacularly failed in the therapeutics. And during the course of my lecture, I will try to give you some ideas of what are the differences and what we are probably dealing with. And again, please be skeptical, because at that time in our knowledge, we don't have too much of direct data of what happens to glial cells. in the brains which are damaged by COVID-19. However, we have to look into the future and we have to design, obviously, the routes for investigation. So what is, I think, important to know that this is not the first pandemic and most likely not the last pandemic. And there were many viral pandemics in the history of mankind. Probably the first one was described by Livio, which happened in 412 AD. And since probably the end of 16th century, many of these epidemics were connected to neurological and neuropsychiatric sequels. From the very beginning, these epidemics were mentioned in Germany when it was somnolentia, for example, or lethargic illnesses in Western Europe. In London, that was an epidemic of febris comatosa, then malmazzucco, which is very, very nervous form of influenza. And mazzucco, I think, means stubborn in some Italian dialect. Schlafkrankheit in Germany again, coma somnolentium, finally a big thing of nona, which was again a very nervous form of influenza in Italy at the end of 19th century, which was associated with infectious syndrome, which characterized clinically by lethargy, ocular pulses, and a febrile state. And anatomically, it was defined more or less as diffuse encephalitis. Few people survived the nona, but those who survived had neurological and neuropsychiatric symptoms till the end of their life. Finally, the previous major influenza epidemic, the Spanish flu of the beginning of 20th century, ended up with encephalitis lethargica or von Economy encephalitis, which raged through Europe for about 15 years, killing probably at least 1.5 million of people, although we still don't know whether it was indeed linked to influenza virus, but nonetheless, it followed it immediately. And of course, now with this context of this COVID pandemic, and which has a rather malignant clinical presentation, we also have to think about neuropsychiatric sequel of that particular disease. So before going into discussing what happens to glial cells in COVID, let me do a very, very short introduction of what glial cells are. And of course, our brain is a network, and this is an absolutely outstanding network which produces an incredible computing power by connecting about 200 billions of cells with probably at least 100, if not more, trillions of connections. And that gives our brain many petabytes of memory and many exaflops of computing power, and all that at an expense of mere 12 to 16 watts per hour. So this performance is, of course, unmatched by any computer in the world, or even by all computers taken together. They still cannot do what our brain does at that particular expenditure of energy and in real time. And in order to support that, there are several brain organ-wide networks. First of all is, of course, connectome. And connectome, these are connections between all these billions of neurons and connections between all these trillions of connections. Then is, of course, vasculature, which provides oxygen and glucose to the brain. And there is, of course, a brain fluid system into which brain floating, and this fluid system is very important for collecting the waste and for washing up the brain interior and exterior. And all these brain-wide networks are supported by specific type of neural cells, which are called neuroglia. So what is neuroglia? By definition, it's a homeostatic and defensive cells of the nervous system. And this is a general classification of neuroglia, which obviously is divided into peripheral nervous system glia, which covers Schwann cells, which are myelinating cells of peripheral nervous system, and which also provide for support for axons and sensory synapses and motor neuromuscular junctions. They're satellite glial cells of ganglia, and this is a huge realm of enteric glial cells which are sitting in the gut. In the central nervous system, the glial cells could be divided into macroglia, which are of ectodermal origin, which are of neuroepithelial origin, which again could be divided into astroglia and oligodendroglia, astroglia being the chief homeostatic cell of the central nervous system, and oligodendroglia being the chief myelinating cells of central nervous system, and of course a big family of glia of special sensory organs of hearing, of taste, of vision, and so on and so forth. And in addition, we have a microglia, which are not strict to sense neural cells because they are invaders. They are science of fetal macrophages, which enter neural tube very early in development, but then they undergo the most amazing metamorphosis. They change their morphology completely. They acquire morphology which has some sort of neuron-like appearance with a small cell body and long and ramified and moving processes, and they acquire major receptors which are characteristic for the cells of the nervous system. So microglial cells are neural cells of non-neuronal origin, which are participating, of course, in brain physiology by taking care of debris in development, by shaping synapses, synaptic pruning, synaptic stripping, and so on and so forth, and which are also chief elements of the defensive system of the central nervous system. So traditional image of the brain is like that, and you can still see it in many animations and in many websites. These are neurons through which sort of electrical impulses are coming. Of course, there are no electricity involved in these impulses unless electricity associated with electron movement through the ion channels. But nonetheless, the reality is completely different. The reality is that there is a symbiosis between neurons and non-neural cells. So astrocytes are covering capillaries, astrocytes are covering neuropile, there are microglial cells, processes of which are going everywhere, there are oligodendrocytes and oligodendrocytic precursors. So indeed, there are two different parts of cells which are making the whole. And of course, it's impossible to say what is better, what is good, what is secondary, what is primary. They are all equal and they are all together, working together, working in concert, making our brain what it is. There are, of course, lots and lots of mythology in glial research, and mythology number one, which probably almost everybody of you have seen once or more times in your life, is that there are 10 times more glia than neurons in the human brain. This is a wrong statement. The numbers are more or less the same. Of course, there is a regional variability. With that comes another sort of mythology that astrocytes are the most numerous glia in the brain. This is again completely incorrect. If you think about it, probably the most numerous are oligodendrocytes, because in human brain, 53% of the brain is a white matter. Nonetheless, it's important to remember, and I think that would be nice to keep in mind. Nonetheless, what does happen in evolution, and that is a big change, is a growth in number, in complexity, and the size of astrocytes in human brain. And here you can see a comparison of human astrocyte and mouse astrocyte, and you see the human astrocyte is immensely more complex, immensely larger, and that translates into the number of synapses which are covered by single astrocyte in our brain. So the rat or mouse astrocyte covers 20, 30, maybe 100,000. The human protoplasmic astrocyte covers up to 2 million of synapses within the gray matter. On top of that, human brain contains a specific types of glial cells, specific types of astrocytes, which do not exist in any other animal except of highest primates. Examples being interlaminar astrocytes, somas of which are sitting very close to pia, and they say long processes which are going through several layers of the gray matter, therefore interlaminar. And there are other astrocytes, again, with very long processes, which are called varicose projection astrocytes, which are also found more or less exclusively in the human brain. So all this is really important to remember, because when we are trying to use rodent models to model human diseases, we always have this problem. These are different brains. Now, there is still a fundamental difference between neurons and non-neuronal cells or glial cells when it comes to pathology. When it comes to pathology and lesion count, what neurons do, they become stressed, sometimes they die, sometimes they survive. They cannot fight really this lesion on the systemic level. They, of course, have certain defensive mechanisms against reactive oxygen species and so on and so forth, but those are confined to a single cell. When you look into glial cells, they demonstrate a completely different response. They change. They change their morphology, they change their biochemistry, they change their physiology with one single aim. They try to fight the lesion, they try to protect the brain. And if they succeed, then the disease doesn't develop or is resolved rapidly. If they fail, then disease develops and the brain ultimately dies. Neuroglia pathology start to receive attention only more or less during the last 10 years, despite the fact that the status of glia as an indicator of a pathology in the brain has been known since 19th century. But it was considered for many, many years that what glial cells do, they have a sort of universal stereotypic response, which is known as reactive gliosis, or it's known as activation of microglia, which of course is a very wrong term, so it's a reactive microgliosis. So for a long period of time, it was believed that the only thing these cells can do, they can proliferate, they can boost, they can become phagocytes, and that's about it. And they are more or less detrimental to a neuropathology. Now we know that this is very, very different. And in fact, reactivity represents only a minor part of pathological changes which are happening with neuroglia. These are changes which are happening in response to direct lesion to the nervous system, be it mechanical lesion like mechanical trauma, or chemical lesion like neurotoxicity, or biological lesion which is caused by pathogens. This causes a response, an acute response, which is reactive gliosis. At the same time, there are many other pathological reactions which are associated mostly with changes in the function of those cells. It could be loss of functions, it could be gain of functions, it could be pathological remodeling, and of course it could be a direct damage. It could be degeneration of these cells when these cells die and therefore lose their supportive and defensive capabilities. Reactive gliosis. So again, we are coming back. Reactive gliosis is not equal to neuroinflammation. So brain, as well as any other tissue, has a stereotypic response to lesion, which is called inflammation. In this situation, neuro is just topical. It shows where it comes. In many, many cases, what we call reactive gliosis is not associated with neuroinflammation and would not respond to drugs aimed against inflammatory process. So reactive astrogliosis, by definition, is an evolutionary conserved, primarily defensive response of astrocytes to pathology aimed at preservation of the brain tissue. There are hundreds of phenotypes of reactive phenotypes. They are age-dependent, they are context-dependent, they are disease-dependent, and recently a very popularized idea about dichotomy or binary phenotypes such as good, bad, A1, A2 is a clear-cut misleading thing. And I really suggest you read this paper which appeared in Nature Neuroscience recently, in which a big group of prominent gliologists, about 90 of us, made a consensus statement on reactive astrocytes. What triggers reactive astrogliosis? Hundreds of different molecules of different origin. They come from synapses, they come from neurons, they come from microglia, they come from blood, for example, when BBB is compromised. So there are many, many different things which are converging in astrocyte and which trigger different pathways which lead to appearance of those disease-specific and context-specific reactive phenotypes. When astrocytes become reactive, they start to send instructions. So they send many, many, many tens, if not hundreds of molecules, which are, again, mostly aimed at reparation, mostly aimed at preservation of homeostasis of nervous system and or regeneration of nervous system. It has to be also clearly understood that the major function of astrocytes is to make barriers. And end feet of astrocytes make glia limitans. They make it in normal conditions in physiological brain, against vessels, against pia, but they also do so against the scars. So astrocytes don't make scar. Scar is made by fibroblasts. Astrocytes make a parenchymal barrier which delineated the scarred tissue. Astrogliosis is not really classified, but nonetheless, we have certain general features which are observed in a pathological brain. And first of all is morphology. So most of astrogliosis is isomorphic. Astrocytes do not proliferate, they do not move, they change their morphology, and then when a pathology is resolved, they come back to healthy state. Only when we have a real trauma to the brain, then astrocytes are coming to this particular area, they delineate it with palisades, and they control making of scar by fibroblasts. Microgliosis, this is exactly the same. So there are many, many different types of pathological microglia, in which a fully reactive microglia would be a phagocyte, would be a macrophage, which is very rarely happens in the brain. In most of the cases, neuroglia never comes to this particular stage. Instead, there are many different pathological forms of microglia, which we still don't know how they are interacting with the brain on a short or long feature. So, systemic inflammation. Neurological presentation of COVID-19, sorry for the typo, are very, very common. So there are many, many different reports, and this is a report made 1 year ago, in which about 62% of patients with severe COVID-19 represented neurological pathology, of which most was ischemic stroke. But at the same time, a very sizable population of them had neuropsychiatric symptoms like delirium, sleep disorders, hallucinations, you name it, which clearly demonstrates that the virus does affect the brain. How virus can affect the brain? First is, of course, direct neurotropism. And I'm not going to talk about neurotropism at all, because the lecture of Roxana is fully dedicated to this particular pathway, and I leave it to her. But there are other factors which, being a product of COVID-19, directly affect the brain. And these are, of course, ischemia, because it's a respiratory failure, that's not enough of oxygen. There is thrombosis, which causes strokes, which causes local ischemia. And this is systemic inflammation, in which lots and lots of pro-inflammatory factors are coming to the blood. And this systemic inflammation is probably one of the most important pathogenetic moments which are contributing to long-lasting neuropsychiatric consequences. So that's more or less how the brain looks like. So we have obviously brain vessels, arterioles, capillaries. This is arteriola, which have an endothelial blood-brain barrier sealed with tight junctions. There are pericytes, and there are basal membranes in between two of them, endothelial, parenchymal and vascular basement membrane. We have a perivascular space, which is really important for example, lymphatic. And clearly, this perivascular space also contains cytokines and sometimes pathogens. And as long as blood-brain barrier is intact, what happens? Astrocytes become reactive. They change somehow their morphology, and they enhance the barrier. Now, sometimes the blood-brain barrier is compromised because of an action of very, very many different molecules, into which I don't have time to go. But nonetheless, in this situation, we have, of course, a very different conditions. Astrocytes start to be damaged, and we have a cytotoxic edema. And then, of course, it culminates in making an abstract and making a brain abscess, in which pus and germs are sitting in the very middle of it, and around it, the barrier starts to be made. And again, astrocytes now move towards this emerging scar. They make a barrier, and they instruct fibroblasts in order to make a proper scar, which would save the brain against this focal lesion. Now, what happens in COVID-19? And of course, one of the most important part of it is what is commonly known as a cytokine storm, which is a hyperreactivity of immune response. And in this situation, many, many different things happen. Blood-brain barrier becomes leaky, cytokines are entering the brain, and this again triggers all these defensive responses of glial cells. They are trying to defend the brain against the pathology. And this response is very different between young people and old people. Why is that? Because in an old brain, we do have already compromised glia. So number of glial cells is reducing. Many of glial cells in adults in an old brain become dystrophic, which is particularly very characteristic for microglia. So in old brain, about 40% of microglia disappears or become dystrophic, so they are functionally incapable. And astrocytes are shrinking with age. So these cells are losing their defensive capabilities, and this probably is what makes a brain of old people much more vulnerable to COVID-19. What happens with microglia? Microglia does all sorts of things. So again, they become reactive. They are trying to fight. They are trying to secrete different factors which are increasing the brain defense. They are trying to repair blood-brain barrier. And of course, if they succeed, again, nothing happens and brain escapes unscathed. However, if they fail, then we clearly have a brain pathology. Even more than that, all this cytokine storm can probably also influence even the brain in utero. And this is, of course, known that very many different viral infections are sort of risk factors for autistic spectrum disorders or for different neurodevelopmental disorders. We don't know whether COVID does the same, but we have to be prepared and we have to look into it very, very carefully. So for the time being, there are a number of neuropsychiatric diseases which received a direct confirmed link with COVID-19. And these are, of course, depression, major depressive disorders, their sleep disorders, their reactive psychosis, and post-traumatic stress disorder. There are several other diseases which did not yet receive a direct confirmation that they are associated with COVID-19, but they might be. So again, we have to look into all this. However, what is really important about COVID-19 is that a part of acute presentations, which are more or less characteristic for all sorts of systemic inflammation, be it sepsis, be it any bacterial pneumonia, or you name it, in COVID-19 we have a delayed consequences, delayed sequel, which are presenting quite a serious neuropsychiatric landscape. They are manifested by delusions, by hallucination, by disorganized thought, by foggy thoughts, by many symptoms which are not really similar to a normal classical major depression after infectious diseases or systemic inflammation. And in many cases, those sequences and sequelae are resistant to a therapy. And probably we have to think about making a new definition, which would be a post-COVID-19 neuropsychiatric disease. And here comes, of course, the speculation what happens. And that, of course, look at it as a speculation, nonetheless, that we have a normal glia, a normal healthy brains, and systemic inflammation comes, and we have prevalence of reactive gliosis, which is protecting the brain. But then after the end of this particular period of acute COVID disease, all these cells have to go back to their healthy state. And it might well be that in this situation, this particular return to a healthy state is disturbed, and this glia enters a new state, an aberrant state, which lasts for a very long period of time and which does affect the brain from the point of view of cognitive capacities and as well as neurological symptoms. So here we are coming to the very end, and this is a summary of my talk. That COVID-19 does affect nervous system and is often accompanied with acute symptoms. However, it also has a delayed symptoms, which could be defined as neuropsychiatric post-COVID-19 syndrome. And damage to or aberrant recovery of neuroglia, and this is not a chronic neuroinflammation, may contribute to this neuropsychiatric sequel. And on top of that, SARS-CoV infection and COVID-19 may boost also neurodegenerative diseases, which are, of course, all linked to ischemia, to cytokines, to aberrant microglia or aberrant astroglia or reactive gliosis. And that is very likely that we may see consequences in terms of number of people suffering from neurodegeneration. And I'm coming to an end, and thank you very much for your attention.
Thank you, Alex. Thank you very much. So I'm checking I forgot to tell you at the beginning that we have a question and answer box dedicated to the questions. I think that maybe Jonathan, the Secretariat, mentioned this in the chat. Obviously, you can also use the usual chat box. I think that someone did that, but it would be more convenient to me if you use the questions, the Q&A box for the questions, so I can go directly there. Anyway, I think we have a question in the chat box by Nanu Panu. Jonathan, should I read the question? Should the audience ask the question themselves?
Yes, if you could read the question, that'd be great.
Okay. So this is the question for Alex.
Yeah, it's asked me.
Can you see it? I mean, the question is, is anosmia in patients with COVID-19 associated with brain damage?
Well, again, I think it would be much better if this question would be addressed to Roxana, because she is going to overview the roots of the virus into the brain. And one of those roots is, of course, through the olfactory system. And it is very, very likely that by using this transolfactory route, the virus is infecting these neurons, and these neurons may respond by simply shutting themselves down. And this is olfactory system, which is very, very peculiar system for which new neurons are constantly born. So even if you lose all your olfactory neurons for a while, after several months, they will come back from subventricular zone. So yes, it probably reflects brain damage, and there are some ideas that this might be a defense of the brain. So by shutting down these neurons and by killing these neurons, you shut down this particular route. But again, I think that Roxana will definitely go into this in much more details.
Okay, it's raining questions now, Alex. So open your umbrella.
Okay.
Okay. Next one is by Hendrik Denius. Is there any evidence for this aberrant glial state you mentioned? And this, in a way, it's something that I want to also to ask you, because, for example, there are neuroimaging techniques using PET tracers like PK, 11195 that can bind to activated microglia. So is there? This is a very good question. Do we have actual evidence?
Yes, absolutely. And we have this evidence even for influenza. So there were a couple of papers published by Japanese researchers in virus after influenza of 1979, in which they found lots of aberrant form of astrocytes in this brain. Mostly that was plasmatodendrosis. This is really direct damage to astrocytes, which leads to them losing their capacity to control the brain and to defend the brain. So yes, from other diseases, we do have evidence of aberrant glia in the aftermath.
Okay, thanks. So Irina Rafil, she's asking, did you see that? Okay, I'm recapitulating. So considering possibly affects disease like schizophrenia and bipolar disorder, there's much current knowledge research on how it affects neurotransmitter release plus function?
Well, I mean, obviously, all these diseases of cognition and mood and the brain are associated with aberrant neurotransmitter homeostasis. Now, as far as glia is concerned, in almost all of these diseases, there is one converging molecule, and this is glutamate transporter. So as you know, about 70% of all neurotransmission in our brain is made by glutamate. And neurons do not produce glutamate, so they rely on glial cells for catabolism of glutamate. So glia removes glutamate, then makes glutamine and supplies neurons with glutamine. So without astrocytes, it doesn't work. And glutamate transporter is a central molecule for it. And if you interfere with that, then obviously you potentiate excitotoxicity and you potentiate aberrant neurotransmission. Now, what about COVID-19? We don't know. But in many, many diseases, including systemic inflammation associated, for example, with sepsis, there are evidence for injuries to glutamate transporters and glutamate shuttle.
Excellent, thanks. How about COVID-19 and the enteric nervous system?
Don't know.
Okay.
Well, I mean, again, we can only speculate. So clearly there are gut presentations of COVID-19, so clearly it can go into enteric nervous system and may do things. So yeah, I mean, but I don't know.
Okay.
No.
Are there any? No. So you're answering the one by Rodrigo.
Yes. No, there are no evidence because we don't have it now. To be very honest, the situation so far is very, very controversial. So post-mortem examinations in many cases do show areas of gliosis. In many other cases, they do not. So that is probably a heterogeneity of the disease. Now, the only thing I would like to tell you, Rodrigo, please don't use the term microglial activation. Microglial cells are activated every second of their life. What we are talking here is reactivity.
Okay, I'm going to read the other one, but then we stop. Because otherwise, you know.
Yeah.
So let's see. I'm getting lost here. So can neuroglia be targeted for treatment of obesity?
Well,
wow, this is a tough one.
Well, I think it's slightly outside of the topic of our webinar. And yes, there are many, many evidence that astrocytes and microglia and hypothalamus do suffer in obesity. So probably they could be a target, yeah.
Okay, so now we have two questions by Anna Maria Zagran. Hi, Anna Maria.
Yeah, Anna Maria, nice to see you.
Yes.
Nice to
read from you. I'm not seeing you, Anna Maria, but we know you are here. So what could enable the recovery of post-COVID aberrant glia? And the second part is also, do you think the aberrant glia could impact on a long-term the neurovascular coupling?
Absolutely. Yeah, absolutely. I think that is probably one of the very, very likely outcomes, which are, so in a sense, all these aberrant changes probably are going to shift completely the homeostasis of the nervous system. And I think this is indeed, how to say, decisive point, whether it goes pathological route or whether it comes back. So in principle, that bifurcation, if you will, after the end of the disease, whether it goes to a pathological glial state or brain state, or it returns to physiology, this is the most important, yeah.
Okay, Bianca, do you think the difficulty of getting oxygen to the brain because of COVID is the reason why many glial cells and astrocytes are dying? Yeah. Okay. Is gliosis, reactive gliosis reversible? I think you mentioned this.
Well, we don't know, but in most of the cases, reactive gliosis is reversible. And try to think about it. We still live about 100 or 70 years with a healthy brain. So despite all the exosomes, despite all the exposure, despite all the pathologies and everything and stresses, our cells are coping very, very well. So in principle, there are very few, if at all, pathological cells in normal brain. Pathological cells means that something really went wrong.
Okay, let's try to answer the last ones, because I'm really glad that we are having so many questions. So this is a very good sign. So COVID-19 and blood-brain barrier. What could you say about that?
This, I think that she will just...
Okay. Yeah. Maybe the last one, and then we really have to switch to Roxana. Any links that have been associated thus far between genetic polymorphism and the aberrant gliosis? Who knows?
I don't know.
Yes. Okay. And that's the best, I mean, most honest answer a real scientist can give. Absolutely. Okay, no, thank you very much, Alex.
Thank you, my pleasure.
Yeah, stay with us, of course, because
of
in
course
the end, maybe, Roxana, yes, but I'm just reading very bad news here. Jonathan, nothing from Roxana yet?
No, unfortunately, not yet.
Oh. Okay, this is really unfortunate. So, okay, at this point we have to jump to Livio. Yeah, we are changing the agenda. Okay, well, yes, Roxana Carare was supposed to be the first speaker. She's having some problems with the connection, so we are really hoping that she will make it before the end of the webinar. So in the meanwhile, at this point, we go to Livio Provenzi. The title of Livio's talk is the hidden pandemic, psychobiological footprints of pandemic-related stress in pediatric populations. Here, the focus is shifting a bit. Livio is going to talk to us about not just the direct effects of the virus, but the psychiatric, psychological repercussions of mostly the social restrictions that we all are experiencing, but that have been impacting more dramatically on the young population. So, please, Livio.
Thank you very much, Fabio. You see the slides, I guess, and you hear me. Yes, as Fabio said, my talk is going to be on the same topic, but on a different side. So I'm not going to talk about the consequences of COVID-19 disease, but I'm going to talk about the consequences of the healthcare emergency as a global traumatic experiences. So from a stress perspective. And I'm going to do this by focus on pediatric populations from pregnancy to adolescence. And I would like to focus to center the topic of my talk on this metaphor of footprints, because I think that what Alexei with this brilliant presentation started already to do is saying that COVID-19 is not something that is transient. Yes, it's going to be transient, and we hope it's going to be transient, but of course it's going to leave some kind of footprints, and we can see them in the shape of the neural consequences or the glia consequences of COVID-19 disease, but we can see them also in a different shape, and I hope that my talk is going to clarify this. I would like to start with from here, from this place. We are in New Mexico. Last year, White Sands National Park, and here is the place where paleontologists just discovered a set of footprints from 2 adult individuals from Pleistocene. And based on the shape features of these footprints, they were most probably escaping from mammoth or giant sloths or giant animals anyway. And what is most interesting of this discovery is that they also found out a 3rd set of footprints. You're going to see it now. And these were smaller footprints, probably of a young child. So this was probably a family, and they just let the child step on her feet for a few seconds as they adjusted for proceeding with a hopefully successful escape. So we can see that our footprints tell us who we are, and they tell us who we are since Pleistocene. And what these footprints tell us is that we are animals that care for each other, and we are animals that care especially for babies and for children. Our caregiving nature is probably emerged together with bipedalism many time ago. When our ancestors started walking on 2 feet, we know that the space in the maternal womb for full brain growth decreased dramatically. So our brain maturation had to end outside of maternal womb after birth in the external caregiving environment. And this is one of the reasons why, contrary to this baby giraffe, our newborns are not capable of walking few hours after birth. And we know that even, in Italy, we become independent after something like 30 or 40 years of life, but this is another story. But the long-term advantages of these, of having a brain that hence its fine-tuning after birth is that the brain complete the maturation, embedding social interaction, so embedding social learning into neural wires. So our neuroplasticity is a relational and social phenomenon. And this is so true that in smarter primates, we know that the time spent caregiving is directly linked with the IQ of parents. In other words, we can say that we are born to be wired. And this is especially demonstrated by, for example, neonatal imitation. You can see here some examples. You have a young macaque, few hours after birth, and you have a newborn. And what is really striking is that this newborn is going to imitate tongue protrusion, even if she doesn't really know to have a tongue. On the left side, you see a classic experiment from developmental psychology. It is called the still face procedure. Here you see the child and the mother, they are playing together normally with no use of toys. And at a certain time, we ask the mother to become unresponsive. So not talking, just looking at the baby with no smiles. And you see that the child immediately catch that something is wrong, avoid the gaze. And then you're going to see that she tries to re-engage the mother by smiling. She points in the environment because she knows that this is going usually to activate the mother, but nothing happens for about 2 minutes in this task. So she reaches out, and then you're going to see that progressively, this child is going to say, okay, come on, what is happening here? And she starts to lose the control of posture. She starts to make stress signals. You're not hearing the sound, but she's making some screechy sounds, avoiding the gaze again. So you see that we are not only born to be wired, but we are also highly sensitive to interactive and relational raptors. And of course, in this task, we let the mother re-engage with the baby after 2 minutes, so everything goes back to normal, hopefully. And another thing that we are starting to understand during the last 10, 15, or 20 years is that of the emerging field of behavioral epigenetics. The classic work by Moshe Zif and Michael Meany has shown that the effects of early social interaction with our caregivers, they are so important that they may contribute to the long-lasting regulation of the epigenetic machinery in animal models, and especially by changing the stages of DNA methylation in stress-related genes. And now we know that this is happening also in humans. For example, we know of methylation changes in these target genes in children born by mothers with prenatal depression or anxiety, preterm infants exposed to high levels of pain, children with a history of maltreatment or abuse, socioeconomic disadvantage. But we know that methylation is also sensitive to other types of environmental adversities. For example, acute traumas like the ice storm in Canada in the 1998 and the 9/11 World Trade Center attack. So suddenly, the COVID-19 virus arrived, you know that, of course, and these are actually unprecedented times, even if Alexei already showed that we had previous epidemics in history. But never before like this virus has completely disrupted our social wires. And this is just an example, a sample of the Time magazine covers that COVID-19 deserves, 16 covers in 18 months. And I would like you to think that this just happened in 1 year and a half. And the record man for the number of Time magazine covers is Richard Nixon with 55 in a life, and Hillary Clinton for women, 23. So it's really something that completely impacted our life, our time. And so we started to wonder, okay, how is this traumatic experience, this global trauma, leaving footprints in our relational and psychological life? And we actually started chasing these footprints, and we started to do this in a life course perspective with multiple projects, from pregnancy and birth to children with disability and their parents, and to the mental health of adolescents in community samples. And I'm going to present you the findings of these studies that were carried out at the Scientific Institute Mondino in Pavia in North Italy. So the first one is the Mamco Project. In this project, we enroll 10 neonatal units during the first lockdown period, so back to April, May last year. And our aim was to follow up the mothers who were pregnant during this period and their infants, excluding actually the mothers who were tested positive for the SARS-CoV-2 virus. So just those who were exposed to the pandemic on the stress, so what we call pandemic-related stress, not just the virus itself. And then we started to follow up the growth and the development and the mental health of these mothers and these babies from birth to 12 months with a regular pace, so 3, 6, 9, and 12 months. And at birth, we also collected buccal cells to analyze and to estimate the status of methylation of specific stress-related genes by using next-generation sequencing. And what we are trying to discover is that, first of all, there is an increased high risk of depression and anxiety in these mothers, and this is related not just to the exposure, to the actual exposure to the SARS-CoV-2 virus, but to the exposure to pandemic-related stress. So mothers who reported retrospectively higher levels of stress during pregnancy also had higher risk of clinically relevant elevations in anxiety and depression. And I would like to stress the fact that these rates of depressive and anxiety symptoms were well above the percentages and the rates of pre-pandemic community samples. Interesting, we also found out that in the neonatal unit that invested in follow-up home visiting programs during the pandemic, these mothers from these units had better mental health at 3 months, and basically they had no elevated risk for parenting stress and other forms of mental health problems like depression and anxiety at 3 months after delivery. And this is a paper that is in progress. And then last month, we just published this path analysis paper where we show that there is actually a sort of cascade effect. So starting from the exposure to adverse conditions, prenatal COVID-19 related maternal stress, and prenatal reduction in maternal social support, which is another source of stress for these mothers, these two factors were significantly associated with postnatal maternal state anxiety at birth, and these in turn was significantly associated and predictive of postnatal maternal parenting and postnatal maternal bonding or feeling of attachment to the infant. So you see that we start to see a movie going on, starting from pandemic-related stress to an altered caregiving environment. And what we are starting to understand is that this alteration in the caregiving environment and maternal mental health is going to ultimately affect the behavioral well-being and behavioral development of these infants. We see, for example, that these infants at 3 months may have greater risk of difficult temperament, especially in the regulatory capacities. And again, talking about footprints, you see here, this is a paper that has been just accepted last week. You see that this effect may be at least partially mediated by an alteration of the methylation status of the serotonin transporter gene, the SLC6A4 gene, especially in the promoter region, where we measure methylation at specific CpG sites. You see that higher pandemic-related prenatal stress was associated significantly with increased methylation, and increased methylation was predictive of more difficult temperament at 3 months. So going on with this life course trajectory, we then focus also, as I said, on children with disability and their parents. And we launched a study which is called the Enforce project, because suddenly with the lockdown, the therapeutic actions and the rehabilitative programs of the children was suspended, at least for a brief period. So we wanted to understand how this suspension was affecting the mental health of parents and the developmental trajectories of these children. You can see here that the great source of problems and worries for the parents was indeed not just worries about the COVID-19 infection, but the suspension of the therapeutic and rehabilitative programs of these children. And these worries were the only one predictor of anxiety and depression in parents during the lockdown. So we started a telemedicine approach in order to provide continuity of care to these families and to these children, responding to their needs, the rehabilitative needs, but also the actual needs related to psychological stress during pandemic. And you can see that families reported benefits for the child and benefits for the parents, more than 80% of families. Again, we focused on the effects of COVID-19 lockdown on the progression and on the well-being of children with language problems. And these as I said, these telemedicine interventions are very important because during this unprecedented healthcare emergency, children rehabilitation achievements may be actually lost or impaired in some way. So this is something that we actually observe, for example, in children with dyslexia, as more than 50% of them had decreasing reading speed and higher reading errors from before to after the lockdown period. Apparently surprising finding from patients, pediatric patients with migraine, because during the first wave, the first lockdown period, we discovered that children with migraine reported better health status during the lockdown with reduced intensity and frequency of the symptoms. And this is probably a side effect of the reduction of social contacts and social relationships that may be a source of stress for these children, for example, leisure activity, sport, and school. And so this further supports the notion of migraine as a clinical condition with a strong psychosomatic component. Nonetheless, the general health of adolescents greatly worsened during these last 18 months. And this is something that has been made clear by recent publication in Lancet. And we also found in a study led by Dr. Martina Menzi in our institute that 80% of a community sample of adolescents of more than 1,200 adolescents, 80% reported acute or post-traumatic stress symptoms, and suicidal ideation and attempts largely increased from 2019 to 2020. So we can ask ourselves, where is chasing the footprints of the pandemic-related stress leading us? What is teaching to us? And I would like to focus on at least three major implications. The first one, by promoting, I think that by promoting and sustaining this kind of psychobiological research, we really be able to shed new light on a hidden pandemic, but yet critical footprints of acute and chronic adverse experiences. We should learn from the present pandemic, and we should look for the markers of unavoidable biological learning that occurs in our body. Likewise, the glia change that we can see in COVID-19 infection. There are other biomarkers which are more dangerous, I guess, because they are hidden. There is no infection going on, but there is something, some kind of footprints that are left in our biology, in our DNA, for example. So this research is warranted to unveil potential targets of successful preventive and therapeutic interventions in the future. Second point, I think that this pandemic has really shown how far we are from well-known claims of scientific citizenship. But this is something that has been growing in the last 10 years. And we know the difficulties in delivering science outputs to citizens. This has been a key limitation, for example, of mitigation and containment strategies. And actually, it risks to negatively affect the vaccination campaign nowadays. So I think that at least three actions are needed from, on our side as scientists. And one is improving our ability for reach out skills. The second one is educating citizens to the methods of science, avoiding slogans, and consistently engage patients in research, not only as subjects, but also as co-drivers, and, for example, in defining the objectives and goals of research in a way and in a shape that is consistent with their own goals and interests. And finally, and most importantly, this is something I really care for, I think this is a great opportunity to change our cultural mindset around care. We have faced the pandemic embracing a mindset of war against the virus. Here you have some examples, from acclaiming nurses as heroes to blaming them for some restrictions, from looking to science to discredit scientists, from everything's going well to give back our freedom. And now we see how it's going to be with the Green Pass and the vaccine. And I think that blaming people is not the right strategy. I think that this war metaphor and this war mindset is a typical paranoid response to challenges in human psychological functioning. And now I think it's more time to acknowledge that this metaphor is completely ineffective. It promotes idealizations and devaluations, and it contributes to creating more inequalities and contrapositions. I think that the virus really just made us aware of how much fragile we are. And we need a different mindset, which should be grounded in a culture of fragility to create better healthcare systems. While war calls for aggression, fragility demands for care. So this is the end of my presentation. I just want to acknowledge all the colleagues of my group and colleagues and friends with which I shared this project during the last 18 months and who made these projects possible, especially Professor Borgatti, which is the head of the Child Neurology and Psychiatry Unit, and the colleagues of my lab, Serena Grumi, Elisa Roberti, Elena Capelli, and the students and neuropsychiatry residents from the Mondino Foundation. Thank you very much for your attention.
Thank you, Livio. Thank you very much. So I'm checking the Q&A box. I don't see any questions. Well, we moved quite abruptly from neuropathology and neuroinflammation straight to mental health and epigenetics in children. So it's quite a jump. So that might have disoriented a little bit our audience. I will start with one question that maybe it's a thing that does not apply necessarily to children, or maybe it does, but it's something that is making me reflect a little bit about how social interactions have changed during the past 18 months in the general population. I mean, nowadays we interact with masked people, okay? Other than our spouses or kids at home. When we go out and at work or at the bar or meeting with friends, we've been interacting with masked people. So people hiding a lot of reference points that we use to understand their reactions, to interpret their reactions. So we just interact with people and try to read them just by the eyes, but we do not see whether they are smiling or not, whether they are moving somehow. So we are missing a lot of almost invisible hints that yet are so much important for social interaction. Okay, so what do you think about that? So is this something that may actually impact on social relationships in these difficult times, in addition to all the other problems we are having?
Yes. This is a really nice question. Actually, I know of one group in Italy and one group in the US that is working on this in a healthy population. So the effect of wearing masks on the ability of children, not just adults, but children, to elaborate faces and to give a subjective response, contingent subjective response to emotional expressions, for example. In our group, we are starting a project on children at risk for autism, for example, because we know that they have a very different type of face elaboration. So they have more an holistic elaboration, less holistic elaboration, so more focus on details. So the mask may be really disrupting their ability to enter in social interactions. And since this is going to be something long-lasting, it's not just 1 week, but it's now more than 1 year, we wonder how is this going to affect the emotional development of these children. And there was a colleague, a pediatrician, that wrote to me actually asking me, are we facing, you saw the example of the still face paradigm where the mother starts to have this still poker face. And yes. She asked me, is the wearing mask a big still face on the universal?
Yes. Exactly, that's what triggered the question. Yes.
I don't think so. I don't think so because, of course, we are sensitive to interactive raptors, but we are also able to promote behavioral reparation and the ability to cope with stress. But of course, in the youngest infants, I hope we can have data to say something about that in the future. For what I see in our institute, all the children I see around in the corridors, when I say hi, wearing the mask, they just smile and they interact normally. But who knows what happens, I mean, when you look at the footprints in the brain. So this is something that we should study.
Yeah, but also, that might also depend. Well, of course, they're much more plastic than the adults. But I think that we got much better at reading the interaction just by the eyes, or maybe squinting more than we used to do before wearing masks, in order to show our real feelings. Anyway, now we have questions. Any relationship between or studies connecting cortisol rhythm and the levels of stress?
Yes, this is very interesting because, as you know, stress is usually considered the hormone of stress. We didn't collect cortisol because when we started, we were just lucky to engage 10 neonatal units. It was a period in which we were not supposed to exit from our houses, so it was very, very good to have this network of people involved. And we decided to focus on methylation because it's probably a more sensitive measure of long-lasting effects of stress. But it is true that diurnal cortisol rhythm may be flat, for example, in chronic stress. And but studying cortisol in saliva in newborns is really tricky because cortisol may be affected by a lot of contingent events, and we were not in the neonatal units collecting saliva. We asked clinicians to do that in the different units. So we would have been exposed to a lot of uncontrolled biases. But this is, I think that probably someone is doing that. I am in contact with Martha Welch in New York University, Columbia University in New York. And I guess that she's doing something on cortisol, very similar to our project.
Okay. Differences depending on diet?
I really don't know. A difference in what you mean, on neurobehavioral outcomes or outcomes of stress during pregnancy. I'm not an expert of diet. I have other colleagues who may be better suited, so I don't want to say stupid things on this.
Then there is another question, which is more of a comment than a question, almost a philosophical one, saying how it will be if we think the science of impact of COVID-19 for one giant step in the evolutionary phase of Homo sapiens, rather than always taking it in a negative sense.
Yes.
Well.
Yes. This is this is something. This is really a clever question, I think.
Yes,
maybe I don't know on the evolutionary dimension, but Panindra, I guess the name. I have a colleague from UK that always asks me, why are we always looking for negative consequences of stress? And I do agree on that. So actually nowadays there is a new, very young field in my field of studies that is going to focus not just on the negative effects of stress, but looking at stress from a different perspective. So when we face with stress, we become specialized in coping with stress. We know that there are many findings from this perspective, for example, in animals, in birds. We know that birds exposed to dietary restrictions, for example, they become very small, but in doing this, they are also capable of escaping from predators more easily. So I guess that we should focus on the, let's say, adaptive effects of modifying our biology to cope with stress. We are sometimes too much focused on the negative side, but this is something very interesting. And this is something that I usually refer to by saying not resilience, but biological learning. And other researchers, such as Carolina de Viet from Netherlands, they say, for example, specialization in stress response. So yes, it's a good question.
Okay. And I believe we have the last question by Anna Maria Lagran. Thank you for the excellent presentation. I join Anna Maria in congratulating you, of course, for a really nice talk. What could you recommend to mitigate the effect of pandemic during pregnancy or on your development? Do you think that education and increasing understanding of the epigenetic effect could help?
Well, I think if I had one-shot response to this, I would be very rich, or at least I would send a letter to our Ministry of Health. But anyway, I think that there are many actions that we can do. I just give you a brief example related to home visiting during the pandemic. I think that we can take advantage of this pandemic, at least in my field, so pediatric and developmental psychology, healthcare, by really promoting a new culture of parenting. We need to talk with parents, we need to work with parents, we need to create new forms of partnership between physicians and caregivers, because this can really make space for new ways to take care of children, especially for children at risk exposed to adversities. I don't think at one specific action, but I mean, in a therapeutic way, but more as an opportunity to grow a different culture. And as I said, a culture grounded in fragility and in the value of parenting as a source of prevention.
Okay.
Thank you.
Thank you, Livio. Thank you very much. Okay, so now we are going to the third and last presentation given by Roxana Carare. Roxana is a professor of clinical neuroanatomy, quality and diversity lead at the University of Southampton. The title of the talk is The Neuropathological Complications for COVID-19. Welcome, Roxana.
Hello, thank you so much.
A little bit worried.
I'm so sorry. Many apologies.
Well,
I think it's the fact that I'm stuck between two time zones, and yeah, I'm sorry.
No problem.
That means that, however, on a more positive note, we're going to end with more neuropathology. So I'll just share my screen now and hope that it works. Can you see?
Not yet.
Is anything obvious yet?
Okay. Yes, please.
Yeah, is that okay?
Yep.
All right. So I'm going to put it on full view and hope that it still works. Are we still okay? I'll take that as a yes.
No, I think you have to change the view, because now we are seeing, you know, the small screen on the right upper corner. Okay, perfect.
Is that better? Excellent. Yes. Thank you. Sorry. Thank you. So I think I'm basing the observations or the talk on the observations that my colleagues from University College London have made and have published actually in neuropathology and applied neurobiology. And I have to say that it's one of the best reviews that we have so far with the neuropathological findings in COVID-19. So yes, of course, I'm sure we all know, and you've heard today as well, that in terms of the signs and the findings, the morphological findings post-mortem in COVID-19, there is an acute interstitial pneumonia where there is injury of both the endothelia and the epithelia, inflammation, deposition of fibrin, microthrombi, both systemic and pulmonary, and I'll come back to that because that is also a feature in the brain. There is lymphopenia, depletion of the white pulp of the spleen, and myocarditis, pericarditis, and pancreatitis. In terms of the neurological involvement, there are by far the commonest symptoms are myalgia and headaches, but there are also vomiting, confusion, hypogeusia. We know about the loss of sense of taste, hyposmia, loss of the sense of smell, and like I said, I would come back to it. Cerebral vascular manifestations, which are quite frequent actually, in the form of stroke, venous sinus thrombosis, and overt hemorrhage, intracerebral hemorrhage. There's also Guillain-Barré syndrome, myelitis, encephalomyelitis, and meningoencephalitis. So quite a wide spectrum, and out of which actually quite a few of these symptoms were seen quite frequently here in Southampton. So one of the first questions that we need to answer is how the virus accesses the central nervous system. You may have heard this before. Of course, the most obvious route is the route directly from the nose along the olfactory nerves. But then we mustn't forget the hematogenous route, so via the blood, and the infection of the endothelial cells. Because of course we know that the virus binds to ACE2 and then can cross the blood-brain barrier. There are also some reports that the virus might actually cross from the immune cells in the respiratory tract straight into the lymphatic system or even the blood. And there could be an autoimmune mechanism where autoantibodies against the virus can attack the cerebral endothelial cells. And this latter mechanism appears to also be implicated in the side effects of, very rare side effects of the vaccines. But then again, it's present in probably all the vaccines. So it's just that we haven't had mass vaccination as we have now. We've never had it in our lifetimes. So about the access of SARS-CoV-2 to the olfactory nerves, there's a brilliant paper that's come out last year, actually, in Nature Neuroscience, and they have very convincing evidence of how SARS-CoV-2 antigen is found on the olfactory nerve endings in the nasal mucosa, and this has been shown by immunocytochemistry as well as in situ hybridization, as you have on the right-hand side of the screen. So very quick recap about the molecular side of things and the implications upon the cerebral endothelia regarding these mechanisms. So we need to know, first of all, or remind ourselves of the angiotensin, renin-angiotensin system. So we know that angiotensinogen is cleaved into angiotensin by renin. Now, angiotensin, under the influence of ACE, results in angiotensin 2. And angiotensin 2 is the bad one, I suppose, because it induces vasoconstriction, inflammation, atrophy, and fibrosis through its action on the AT1 receptor. So, of course, we know that quite a few cardiovascular medications are ACE inhibitors because we want to act upon and prevent vasoconstriction, inflammation, and fibrosis. Now, ACE2 is an enzyme that does exactly the opposite to ACE. So what it does is it gets rid of angiotensin 2 by cleaving it into angiotensin 1 to 7, which binds to the MAS receptor. And the MAS receptor, once it's being bound, it promotes vasodilation, reduces the blood pressure, and it's basically very similar in action compared to the AT2 receptor. Now, ACE2, if you look on the left-hand side of the diagram, also cleaves angiotensin 1 into angiotensin 1 to 9, which can then be cleaved into angiotensin 1 to 7 that binds to the MAS receptor by ACE. So ACE2 is a pretty important protective receptor, if you like. Now, in terms of its molecular biology, ACE2 has an extracellular domain that is an N-terminal domain and a C-terminal transmembrane cytoplasm-facing domain, and that's got a tail. The N-terminal contains the protease domain, which actually binds to SARS-CoV-2. Okay, so it's the outward-facing part of the receptor that binds the virus. Okay, now within the brain, the highest expression of ACE2 is found in the pons and the medulla, and that actually might be important because that's where the cardiovascular centers are as well. The ACE inhibitors and the angiotensin receptor blockers, which of course are commonly used to treat cardiovascular disease, result in an increased expression of ACE2 that is protective against hypertension. And of course, at the beginning of the pandemic, people thought, well, if we increase the availability of ACE2, it might bind more virus. I don't think that's actually been proven. Once the virus does bind to ACE2 at the cell surface, we lose the tissue protection conferred by ACE2. And that is, I think, probably the key step in the cascade of events that happens afterwards. Now, normally, the endothelium surface is anticoagulant, antithrombotic, and profibrinolytic, but that's all lost once the virus binds to ACE2. And the reason for that is, it probably takes a little while to take this diagram in, is that as soon as the virus binds to ACE2, then the angiotensin 2 and its receptor, angiotensin 1 receptors, increase. And what does angiotensin 2, if you remember from the first diagram, what does that do? It increases vasoconstriction, so we're losing all the protective effects of ACE2. So we now have vasoconstriction, fibrosis, reactive oxygen species, and an increase in ADAM17. Once ADAM17 increases, that also leads to a positive feedback loop, further increasing the AT1 receptor that acts again upon the ADAM17. So there's this sort of positive feedback loop, and the problem is that once ADAM17 activity increases, that is one of the main drivers of TNF-alpha, which of course is very pro-inflammatory. So TNF-alpha then, along with the systemic cytokines, and I suppose if you add the comorbidities of diabetes or hypertension, that will lead to what clinicians called cytokine storm that happens in the severely affected COVID-19 patients. So it's all to do with the loss of the protective effects of ACE2 and the increase in the vasoconstrictive effects of angiotensin 2 and angiotensin 1 receptors. So what then do we see in the brain? We see an influx of T cells. So normally there are no T cells in the cerebral parenchyma, but in the SARS-CoV-2 infected brains, we see quite a few CD3 T cells. We see an increase in the CD68 positive cells. Now, many people associate CD68 with activated microglia, but they are a marker of macrophages. So if you look on the bottom left-hand side of the diagram, those cells are actually perivascular. So the ones in the parenchyma, yes, they are pretty convincingly microglia, but the ones on the blood vessel are probably just perivascular macrophages that are being activated. There is a high number of T cells and microglia in the medulla, probably because that's where the receptor ACE2 is very highly expressed. And quite extreme and distressing really for the pathologist to see is quite the level of necrosis, the number of macrophages, and these signs of fungal infections. So mucormycosis is a very rare fungal infection, usually associated with very compromised immune systems. It's thought that in this group of patients, it's to do with the large amounts of steroids that the patients have been on and predisposing them to these fungal infections, both mucormycosis and hyphae. So that's what we see now in patients who have died after COVID-19. The biggest problem that I have and my group are working on are the long-term consequences on the brain. And the reason for that is we know that this virus binds to the endothelia. Okay, on the bottom right of the diagram of the picture, you have a diagram of a cerebral arteriole with the endothelium, with several layers of smooth muscle around it, with a leptomeningeal sheet, and with the astrocyte end feet. Now, in patients with Alzheimer's disease, so nothing to do with COVID, we see amyloid beta accumulating in tunica media. So we see it accumulating in these basement membranes that are between the smooth muscle cells of an artery. And summing up about 20 years of research, we know that the brain doesn't have traditional lymphatic vessels, and fluid and solutes are eliminated along these basement membranes surrounding smooth muscle cells. So if these basement membranes are compromised because of an infection with SARS-CoV-2, then this process of perivascular drainage of amyloid and other solutes is probably compromised as well, only it won't be evident immediately. So we may be faced with a pandemic of Alzheimer's disease appearing later in patients that have managed to survive SARS-CoV-2, but that's a hypothesis that remains to be tested. One of the things that we're concentrating on is to try and further characterize the distribution of ACE2 in relation to gender, age, race. We know that there have been reports of quite severe disease in those of Black and other ethnic minorities. The hypertensive status, so what is the exact distribution of ACE2, what is the pattern in hypertensives that are treated or untreated? What happens, because of course, quite a few laboratories have potential for category 2 infection work. We know that the virus has been isolated, so we can infect cerebral endothelial cells to look at the downstream effects. And of course, back to the area of research that is quite close to my heart, which is the pathogenesis of Alzheimer's disease, we need to really determine if there is an increased risk of dementia or cerebral amyloid angiopathy in those that have survived COVID-19. So with this, I will stop, thanking you all for listening to me, and any questions I'd be more than happy to answer. I've put up my email in case there are any comments after the talk. Thank you.
Thank you, Roxana. Thank you very much. So let me see if we have questions. I don't see any questions yet. So I will start with one. I remember I've seen somewhere a couple of papers mentioning the possibility that spike protein might somehow interact with amyloidogenic proteins, triggering, favoring, enhancing the precipitation of either amyloid or synuclein, which might makes the whole situation much more dangerous and scary in the long term. So what's your take on this?
Well, I completely agree with you. I think that it's quite alarming, because the more we understand the mechanism, the more we understand the possible long-term consequences. So yes, you're quite right. I also have seen evidence that the spike proteins interact with amyloid beta peptides and with synuclein. But to be honest, it doesn't take much to promote the aggregation of these proteins that have already started a cascade of aggregation anyway. And it will take decades, as it does in normal sporadic, I suppose, aging and Alzheimer's disease. It will take decades before you see symptoms associated with the pro-amyloidogenic cascade. I think that the problem with we've managed to address the immediate acute problems of COVID-19 through a vaccination strategy. I don't know if this is if we've even scraped the surface of the iceberg of the problems that might occur later on. And when I get very worried when I hear people, and I see another eminent pathologist nodding his head here. I'm so sorry I wasn't there for your talk, but I've read your papers, and I'm glad that you agree here, because I get very worried when I hear people, oh, I'm vaccinated, I had a very easy form of the disease. Yes, that's very good short term. How do we know what the long-term implications are?
Right.
Of the
Go ahead, Alex.
Vaccine. I'm not worried about the vaccine, by the way. I'm worried about
Roxana, thank you so much. And I was really looking forward for your talk. And it's so unfortunate that somehow it didn't work as I wanted, because I tailored my one specifically knowing what you are going to say. And in particular, I think this thing about neurodegeneration and Alzheimer is incredibly important. And one of the things which is just happening now is that we start to learn what beta amyloid is for. And beta amyloid is one of the parts of defensive response to infection.
Yes.
So it goes up and down, and it does so 10,000 times in our life. But if it goes up too much, then probably it can return. I mean, of course, beta amyloid is just one part of Alzheimer's disease, and probably not the causal part. But certainly its accumulation and overaccumulation does take place. But I think it's much more than that. It's really the complex effect of everything, of ischemia, of microthrombosis, and this your beautiful things with mycosis inside. It's horrendous, to be honest.
Yeah. I think the problem is the association with the endothelia, because APP, amyloid precursor protein, is a stress protein. So any infection of any sort will upregulate APP, as you say. We get more amyloid. We know we're not equipped to get rid of amyloid as we would like to, or at least young brains are, but older brains are not. So then you have a protein that actually further enhances the aggregation process in the walls of the blood vessels, and we have ourselves a problem. I'm not even worried about the plaques, because we might all be walking around with plaques of amyloid in our head, but it's the amyloid in the walls of the blood vessels that might create a problem.
No, I mean, this thing with basement membranes is really very important, because this is in essence for lymphatic system.
Yeah.
And somehow, if you start to look into many papers around, people are forgetting that there are more than one basement membrane. Yeah. There is space in between.
There's several.
When we do fixed preparation, it disappears. Of course, we don't see it, but in the brain, it is there. And it's a very intricate tissue. And if you do damage it a little bit, the consequences could be incredible. I mean, I really like this your immunus and everything. Beautiful. Thank you.
We'll have more discussions about this outside.
I would love to.
I can see Professor Blandini getting, you're smiling
Oh, yeah.
at the time.
No,
So
no. Now it's like we happened with Alex before. Now it started raining questions. So
all right, yeah. Do you want me to
yes.
quickly whiz through them? Shall I read them out quickly and answer them as
Yes, probably the best way.
Are there any other factors other than hypertensive status which make BAME at high risk of contracting COVID-19? I think, there may well be, and this is totally understudied, we have no idea what the BAME, whether the distribution of ACE receptors in we hopefully will be able to answer that, whether there is a different distribution of ACE2 in different populations, but I don't think it's just BAME. I think it's also gender, age, maybe. It remains to be seen. What about microvesicles from the virus? Are there evidence in the brain of the microvesicles? I haven't seen them, but it doesn't mean that they're not there. And I think it's just that the neuropathology is still an open field. Unless you look for it, you don't necessarily see it. It doesn't, so it's a very good question, and it's something that we really ought to look for. Do you think that the expression level of ACE2 in the brain, besides that found in the medulla and the pons, could predict the viral infection of certain brain nuclei and explain some neurological manifestations? Well, that's exactly why we want to look at the pattern of distribution of ACE2 to see if there are any areas of the brain that are particularly vulnerable to a high infective load of the virus. People who develop aneurysms after being vaccinated, is that because they have been affected already with COVID-19? I'm not sure there is evidence of more aneurysms. I think there is evidence of hypercoagulability. And as I alluded to briefly before, I was talking to our immunologists here, and I think any kind of vaccine poses a risk of auto antibodies that react, and then you might have an increased risk, but it's very rare. And I think that the only reason that we see the numbers that we see is because we have a mass vaccination, globally, with very high numbers in a relatively short period of time. Link between hypoxia during COVID infection and ACE production. Now that's interesting. I've no idea whether the expression of ACE is dependent at all or interrelated with hypoxia. I don't know. Do you know? Does anybody know? I don't know. I've got people more senior than me here who might know, but they're quiet.
Well, we can't know everything. It's the beauty of life, to be honest.
So yes,
Okay.
I don't know, but that's a good question. I mean, it's something that we could answer probably by, a hypoperfusion model, or, even on a cell in vitro model, and look at the expression of ACE2. Yeah.
Yes, yeah, you're right, Anna-Maria.
Autoantibodies recognizing ACE2. Yes, there are. Yeah, there are autoantibodies against everything. So, yeah.
Okay. And I'm seeing a couple of questions in the chat box. I think that somehow we already answered this question, but nevertheless, I'm going to read them aloud again. So the first one is about the relationship between neurodegeneration and COVID-19. I think that we mentioned this when we spoke about the possibility, that spike protein is favoring amyloidogenesis. The other one is a link with of post-COVID-19 with psychiatric disorders. I don't know if any of you want to comment on this, because we also touched upon this topic already in the previous presentations. So
I think, I don't know. That's a very good question, whether infection directly exacerbates psychiatric illness. That is a very good question. I think that maybe IBRO should consider, long COVID effects, and just actually a study group on this, because it's very pertinent. I'm not aware of it.
Yeah, I believe that this sort of long-term consequences the psychiatric ones are not restricted to COVID. I think that, that these are seen also in post-herpetic infections or in other neuroinfective diseases in the post-acute phase. So it is not something that is restricted to COVID-19. But for all these reasons that we just mentioned, I think that it's better we stay away from this virus as much as possible, which means getting this vaccine as soon as possible for everyone who hasn't had it yet. And now I'm talking to all the people in the audience that are listening or hearing us. Okay, so I think that we are finished with the questions. Jonathan, can we stop right here? Is there anything else left to do or say other than thanking our excellent speakers? I think that this was a really nice webinar with great talks and really nice discussion, lots of very interesting questions. We touched upon 3 different topics which are very well related and yet different one from the other. So I really thankful to Roxana, Carare, Alexei, Verkhratsky. Did I pronounce it well, Alex?
Yes. Well, I mean, KH stands for H, but H doesn't exist in English, so it's all fine.
As an Italian, pronunciation sometimes it's not okay. It's much better with Livio Provenzi. That's really easy for me to pronounce. So Livio, thanks to you as well for a great talk, and hope we will have other occasions to meet in the flesh.
Yes. This is the most important. I mean, I die without Italy. I think everybody.
Yeah.
Thank you.
Okay, so thank you everybody. Stay safe.
See you next time. Thank you very much.
Bye.
Ciao.
Thank you very much.